pleasure to introduce Jos Benoit, a professor in the Department of Biological Sciences at the University of Cincinnati. His research focuses on the integrative physiology of arthropods with an emphasis on stress tolerance, dormancy, reproduction, and sleep, particularly in blood-feeding species like bedbugs and mosquitoes. He has published over 200 scientific papers, more than 70 on feeding arthropods, and has presented at many scientific conferences. Dr. Benoit earned his PhD in entomology and physiology from the Ohio State University in 2009 and has held research and teaching positions at Yale University and the University of Cincinnati. His work has been supported by major funding agencies, including NIH, NSF, and USDA. Dr. Benoit is also a dedicated educator. He has developed undergraduate courses and led international and community outreach programs. He is also deeply involved in a professional service, serving on journal editorial boards, reviewing for journals, and participating in numerous grants and award panels. His mentorship has supported dozens of early career scientists, including postdocs, graduating students, and undergraduates. And with that, please welcome Dr. Jos Benoit, and the floor is yours. Thank you.
Thank you. Okay. Oh. All right, I think everybody can hear me. And so thank you for the invitation today. I've been enjoying my visit so far, even though I got in exceptionally late last night due to, or technically early this morning, I believe, due to a late flight. So I'm going to talk about some of our recent studies on sleep and mosquitoes and how it impacts blood feeding patterns and other aspects along with that. So this is kind of, we talked a little bit about this and a little bit of what is going on actively within the lab right now. The whole seminar will talk about sleep and mosquitoes. We also have projects in tsetse flies and live birth cockroaches as well. And then I'm going to highlight a couple of the other areas. I always get the little snippets of some of the other areas in lab. It's mainly so then technically I can put this presentation on those grants. So no one laughed about that, tried to squeeze it in. So this is going to be the overview. Along with all this work, I was also lucky enough years ago, I spent 4 field seasons in Antarctica with Dave Denlinger at Ohio State. And so that's my photo there. Nothing's better than being able to go collect your bugs on little boats around Antarctica. I would give my left pinky to go back. So, and then I'm going to talk about, I'm going to give the short highlights and talk about some of the sleep processes. So the first thing I'll talk about is a collaborative study with Mike Pollock. He works on fly-mite interactions, and we started the ideas for this grant the moment I was actually hired, and it's looking at ectoparasitic mites, and then it was mainly a combination of Joy, David, Haley, and Mike as part of this project. And we're using Drosophila really only as a carrier to understand ectoparasitic mites. They happen everywhere. If you go collect a whole bunch of fruit flies in this area, you'll find mites on them. It's a pervasive system that occurs in all the different areas. If you get one mite, you're cool. You get a couple, you're probably still cool. You get this many, you're going to die, and they're going to rip you to shreds, and they're going to eat all the pieces. So there's a variation in the level that infects it with the mite fly interactions. And so in here, you can actually select for behavioral resistance. So a lot of the studies we go get natural populations of fruit flies, do prolonged selection experiments to increase resistance to these mites. And we do it, it takes a few generations. In this study, we only selected on the males. And so it takes about 12 generations. If you select on both sexes, you usually get significant deviation after about 6 generations. So it's the added pressure on the 2 different sexes. So we select them for resistance. And then we are trying to figure out what goes on with this. And it turned out, basically through a lot of studies, it turned out we selected on them sleeping less. And so, and the best example I give to you is imagine that mite is about a tick the size of a cat. If you were asleep, it's pretty easy for it to attack you. If you're a little bit more awake and active and moving around, particularly at night, you actually see they're a lot more resistant to it. So, and it turns out we did a whole bunch of studies with this, and pretty much there's a selection causes less sleep during the night, And then we took a whole bunch of known lineages of flies with different sleep levels, and it turns out there's a correlation. The more they sleep, the more infections with the mites. And so that's published, and it was in one of the Nature journals, Biological Timing and Sleep. And so here's a summary for it. If anybody would like to, I put the QR codes for the paper here, and you can click on it and find it. But if you just search my name and fruit fly sleep, this will come up. And so the next one we want to work on moves a little bit to some of our vision work. And this is mainly with Elke Bushbeck and Shubham and Amartya. And so this was a study we did I think it came out earlier in 2025 or 2024, in looking at how osmosis impacts optical alignment of eyes in these cool beetle systems. And so basically the entire summary of this is we work on these sunburst diving beetles. They're great predators of mosquitoes. And so we actually are using them in other studies as predators of larvae of mosquito to evaluate phenotype in mosquito larvae. But what happens in these, it turns out if you mess with the osmosis during development, you actually end up with eyes that don't grow correctly. So if you move them to a slightly higher salt water content, they can't absorb the water into their eyes. So they end up making a bigger lens, a normal lens, but it's on the optics of a smaller eye. So it turns out this happens. And now immediately after this, there's been 3 studies on vertebrate systems that have said osmosis is critical. But as you know, all the entomologists here know the vertebrate people never cite the insect people. And so they never cited our study, and they all came about a year after it and claiming to be the first to show this. So it happens. So, all right, the last kind of little snippet I want to talk about before I move into the sleep is our current studies on tRNA modifications in insect systems. So everybody has learned through the years that you have tRNAs, they recognize the codons, and they make the proteins. All right, that is wildly too simple. So all of these little spots here on the tRNA can actually be chemically modified. And some of the chemical modifications slow translation. Some of them increase translation. Some of them have a higher fidelity. And so there's a lot of different changes there. And so we have a few different studies on this one, but the one I'm going to talk about is we were looking at how the tRNA modifications change during the process of blood feeding in Aedes aegypti. And so we took some previous RNA-seq data and then also looked at modification. It turns out the modification profile changes after a blood meal. You would expect that everybody who's worked on physiology of mosquitoes or blood feed insects, it changes drastically after a blood meal. They're changing their entire protein synthesis machinery, so it has to change. And the one we were really interested for this was tyrosine, and particularly in the fat body, because in the fat body of mosquitoes, that's where they make vitellogenin. And you have to it has a higher proportion of tyrosine than you would expect. And so we actually see that there's an increase in some tyrosine levels, and we expected there would be an increase in the abundance of modifications in the fat body as well. It turns out that that's not true. And so it turned out for this system, at least for the tRNA modifications, there's 2 ways you can usually make more proteins. You either can add more modifications to make it go faster, or you can just make a lot more tRNAs. And they went with we're going to make a lot more tRNAs to make the vitellogenin, rather than higher amounts of modifications to make it run faster. And so it's actually, it's a push to which one is actually more metabolically efficient, depending on the specific situation. So, all right, so that's kind of my short little snippets of some studies. And I always like to stop. Does anybody have any questions on any of these before I move on to talk about some of the sleep studies? Well, for the flies, that's a pretty standard protocol. They have you can use a tracking system, and they consider like it's a 5-minute window of no activity to be the start of a sleep cycle, and then the duration's based on how long that window is. So after that 5-minute window, and then you merge all the 5-minute windows together, then if it's like a 2-hour period with no activity, then the duration of sleep would be 2 hours. And so you can standardly set a number for that for inactivity. And they know that pretty well in Drosophila because then they've done studies where they've disturbed them, that sort of thing. And they know that they're less responsive and that sort of thing. I'll talk a little bit about that with the mosquitoes coming up next. Yeah.
So is that they have less sleeping cycles or that the sleeping cycles are shorter?
For this case, the sleeping cycles are shorter. So they sleep almost the same amount, but it's still slightly reduced, but they're sleeping in these tinier windows. So.
That they are, more of them have none, or overall they have less?
Technically both. But so it's hard to, because of the way the assays are set up, you're trying to control mites that live in this like media and the flies. So like doing the actual abundance number is kind of hard to quantify properly. So it's usually yes versus no. So it's more of yes versus no. So we can count. The problem that you get with them is we try to optimize the assays where they're only getting 1 or 2. Because if you put too many in there, it's a free for all. The mites just start coming out, and then you get 3 on there, then the fly's like walking like this. It's not. It can't get away any better. So then it's like this kind of like just this. It precipitates on itself, and then you end up not getting a lot that are infected. So.
Are they flying away?
It's more of a combination of kicking and these micro bursts of flights away. And so what we have some data we're working on now with it. We think it's the decreased sleep is they look to be like a little bit slower when the mite comes up and touch them. If they're asleep versus not asleep, like a couple milliseconds slower in their flight burst. And it's just enough that the mite can get a hold of their leg, and then it's done. Yeah. So that seems to be what's going on with it. So any more studies on that? Yeah. Any questions? Sorry. Yes, because it's it's a system and how we isolate them. It pulls one versus the other differentially, if I remember correctly. Yes, they'd be nuclear mitochondrial tRNAs predominantly. Yeah. Okay. Okay. All right. No, I'd be happy to answer the question. It's better to do it now because you'll forget it by the end, because that's why I do in seminars anyways. So, all right. So I'm going to move and talk about some of our sleep stuff, which is the main thing from our for the seminar. And so really sleep has really only been effectively studied in very few systems. And particularly within insects, it's really mainly just been bees and fruit flies and a little bit in cockroach, up to the point we got interested in this in mosquitoes here. And it has a lot of complexity to it. And then they generally talk about, there's been a lot of ideas of what is the essential function of sleep. It seems to be different for different organisms. There's usually some trade-offs that are associated with it. No one really necessarily, if you didn't have to sleep, we wouldn't do it. And so it makes you highly susceptible to predation, a lot of other aspects, but there are some aspects that like it's important for recovery, different aspects. But then there's been studies in fruit flies where they have lines that never sleep and they seem perfectly fine. So establishing the exact one, they know eventually in most mammalian system invertebrates, if you prevent sleep long enough, you will just die, like plain and simply. Insects, in most cases, if you prevent sleep long enough, you end up having shorter lifespans. And so it's the same general aspect. So it's the idea is a generally recovery, even though there is trade-offs associated with the state. So, and mainly the trade-offs are like when you're asleep, it's pretty easy to be predated. And so, all right. And then there's been a lot of ones that showing that sleep is important for generally insects. And like I said, it's mainly focused on bees and fruit flies. And so mainly it's ideas like immunity suppressed, behavior suppressed. There's difference in aggression. There's differences in how they learn. So we have some nice studies I'm not even going to talk about here working on mosquitoes. It turns out if you sleep deprive them, they don't learn what a dangerous host is and don't know how to avoid it correctly. So we have some good studies with that, but I'm not even going to talk about that today. So little's known about sleep in mosquitoes and blood-feeding arthropods. So that was our goal of these studies, and it really involved. It really started with the idea is that in all systems, circadian rhythm influences sleep-like states. So, but remember, they're not quite linked together. So it's one of these ones where people always assume they're linked together. You can usually disentangle them where you have circadian rhythms and you usually have a specific time you usually sleep more, but you can sleep outside your circadian rhythms, and there's a lot of different aspects. But in generally, mosquitoes, really great circadian rhythms, so probably have some really great sleep cycles. And so we were interested in starting to look at that. And so it really came down to 4 major points is we did some initial thoughts of just do mosquitoes sleep. We looked at behavioral correlates of it. We looked at some differences between the lines. And then we've now looked at some dynamics between dehydration and sleep activity profiles for this. And so a lot of this work is from Matthew Lewasuin and Clement. And so we kind of have a collaborative R21 looking at mosquito sleep cycles. And so we've been working on this now for about 3 or 4 years. And so do mosquito sleep. So the first thing we did is like, let's search what's out there and write an opinion about it. And we sent a request to Trends in Parasitology. They said, yeah, sure, you can write that. And then they decided to make it the cover of it. It turns out in their bank of mosquito pictures, basically from what we were able to determine, that mosquito is probably asleep. It's a posture. So it turns out they had a picture of a mosquito asleep in the ones that they were allowed to use for their cover. So we ended up going through, and the lines of evidence previously was there were some old studies in the 60s that showed this distinct resting posture that a lot of mosquitoes go in. They lower their legs, they bring their abdomen to the surface and bring their mouth parts. And they're really pressed against the surface in the state. They have really distinct rest and activity profiles. And then pretty much they have all the sleep gene orthologs that have been studied in Drosophila in the mosquito genomes except one. They're missing one that has been studied. And so we looked at some basic surveys of these and just to include within the paper and put it in the supplement of just some postures of Aedes aegypti. And it turned out all the ones we were able to validate a initial sleep-like posture. And so we always call it sleep-like posture, and it's different than their active states. And then we decided to follow up this with another study. And so we want to look at the behavioral correlates of sleep-like states in mosquitoes. And I'm not going to talk about it today. We actually now have neuronal correlates. It turns out in there, if you put some good old electrotenograms into their brains, some parts turn on, some parts turn off when they go into the sleep state. And it looks like potentially some parts that are turning off are associated with host sensing. And so, so we have that, and that'll probably be finished in the next year or two. So this was published in a Journal of Experimental Biology in 2022, and then it got highlighted by them. And again, here's the QR code if you want. They did a nice image. It got picked up from some news sources, and there were some other good images. There's one where there's like a scientist holding up a boom box trying to make loud music to keep the mosquitoes awake. And so it was a fun study. But our really thought with this is initially we just want to look at the behavioral correlates. And if you go and see how this is described in pretty much any review article, there's usually species-specific postures to sleep. There's prolonged periods of increased inactivity, and then there's an increased arousal threshold. So like, all right, we're going to characterize these first, and then usually there's also a sleep rebound. So if you prevent sleep for a period of time, it will rebound in a time when they normally don't sleep as much. Similar to you, or similar to me, who got in late last night, and I slept about an hour and a half later this morning than I normally do. So you see a sleep rebound. And so we did a lot of studies with this, and I'm going to go through it, where we did a lot of imaging, body posture measurements, multiple species, dam systems, videography, and that sort of thing, and combined it all together to kind of get a thorough understanding of the behavioral aspects of mosquito sleep. And so, and we looked at Aedes aegypti, Culex pipiens, and Anopheles stevensii, and pretty much all of them have a resting sleep-like body position. And we've now looked at Anopheles quadrimaculatus and what's the name of the really pretty shiny mosquito with the, yes, And so we've looked at it there as well. And you can see the different, not the pretty side, not that one. It's the one that Woody Foster used to work on from Ohio State. It has like Sabbathes. Yeah, yeah. And so and they have a posture like that too. Weirdly, we should have started with that one. Those pads on the ends of the feet make it actually easier to quantify. But you'll see in every case, their pretty much legs become more parallel to the surface, body position changes, and the head position changes. And then when you actually look at these different metrics, you can pretty much split them into active and sleep states. And they're basically by these measurements, it's the active and sleep states separate the body posture more than the actual species. So it's kind of so they all have a sleep-like posture. And unlike Drosophila, we did some measurements of how long it takes of inactivity to really go into sleep. And we have to add a much longer period than Drosophila. It seems to be at least an hour to 2 hours for them to really be in this sleep state, unlike Drosophila. And it probably reflects differences in their general ecology. They're probably waiting for a host to come by. 5 minutes, it's much longer than the mosquitoes. Yeah. So, yeah. And so we do see when we actually look at these periods of inactivity, we actually then do see, hit the next one, a reduction in responsiveness, that actually less of them will be aroused in response to a host the longer this inactivity period goes. So by about 120 minutes, about half of them become aroused and start moving when a host mimic is added in. So, so then that's the reduced responsiveness. And so then we looked at some of the basic activity patterns based on those metrics. And so, and it turns out, as you would expect, Aedes aegypti, day active mosquito, its sleep is condensed in the night period. No one's surprised by that who works on Aedes aegypti. And then, but then we see the reverse, Culex pipiens and Anopheles stevensii, which are both more active during the night, have condensed more sleep during the day. And so, again, that's not really surprising. Those are two more crepuscular to nocturnal type mosquitoes. And then the sleep amount differs among the different species. And so the total amount of sleep, this right here, may be more of an artifact of partially of lab adaptation. I'll talk about that later. But again, they do differ between species. And so the next one we want to do is the sleep deprivation experiments. So if you deprive them of sleep, can we do it? There's a rebound. We use this little shaker system. And so what you do with that is pretty much you just strap them to this monitor, and then every 5 minutes they get or a little shaking, and it wakes them all up. They move around. And so we have experiments where we either shook them through the whole night or condense the same amount of shaking into a shorter period and then give them a period of recovery. So it's not activity differences within that period. And it turns out that after sleep deprivation, so you have before sleep deprivation, after sleep deprivation in the light phase, they actually sleep more the next day than we would expect. About 20 to 30% more, showing that they do have a sleep rebound after sleep deprivation. And so, but then by the next dark phase, it looks like it's recovered. And so that's pretty much what's been seen in other insect systems and in mice as well. It's that next cycle, they recover, then by that next period, they're in a normal cycle. So we looked at host landing, and so you can see our little host landing assay. You can't see it very well in here. We use these Hemotech feeders, our fake host. We strap on some nice artificial host sweat and stuff on there, warm it up, and then they can land on there, and then you can see the control. And you can't, I don't know if you can really see it that well in here. You go back. I am not going to be able to. Yeah. I'll show the data, but you can see you see how there was a whole bunch flying around there initially. So you'll see them flying around and moving around a lot. And then you see the little dot. I can see it here, but you can't. It turned on the sleep deprived ones. The video's on. They're just not doing anything. they're just like, they're not showing any response there. And then, so, and generally, it actually does impact their host landing. We evaluated at 10 minutes. at 10-minute intervals. If you give it a long enough time, then they do end up being going back to normal. And we think it's just you put that artificial host mimic there long enough that they'll eventually it'll wash out the effect of the sleep deprivation. So, and then we did blood feeding, and the blood feeding is we do live human host blood feeding, which it was me and Matthew, IRB approved. We say that IRB approved. We have the IRB approval, and so it follows what we would expect from the host landing. And so this is a bigger cage that we put them in. We use our feet, and you just pop the foot in there after sleep deprivation, and you just wait to see if they fed or not. And so it turns out this actually does impact their feeding in this short period. So this is the general summary of all of that. We have posture differences in sleep. They have consolidated sleep periods, which you expect. There is a sleep rebound and what you expect in there. And then sleep deprivation impairs host landing and subsequent blood feeding, but it really only impacts the next cycle. And so we have a multitude of follow-up studies between the neuronal part. We have RNA-seq involving sleep deprivation. It turns out sleep deprivation really impacts the expression levels of period and timeless. And so, so we have that data moving along, the neuronal stuff. And then we have some cool studies with like artificial light at night. And then we have some where we actually have outdoor cages slapped underneath a highway to see and it's an area where we see mosquitoes resting all the time. And so we're looking at how actual real life scenarios could impact this. That is because we got some grant reviewers who are buttholes about a review and said, we don't see the real world applicability of this. So we're gonna prove you wrong. So, all right. Does anybody have any questions about that before I move on to the last two parts? It's usually by that next evening, it's kind of gone back to normal. So we really see, we'll do the sleep deprivation, and then we really only see the sleep rebound in about the first half of the next day, which is what you would, Egypti, what you expect. So they're sleeping, you know how a lot of Egypti, I'll show one in a second, how they have that morning activity period, that is gone effectively. So it really suppresses that one. We've tried to figure out ways, and we have some other data where we did the sleep deprivation during their normal active period. It doesn't impact them at all. Like we don't see a rebound then because they're already active and sleeping less during that period. So we have that control as well. I didn't talk about it in the. No, but we haven't even looked at that yet. So, yeah. All right. Any other questions before I move on? Okay. All right. So next, I'm going to talk about our different sleep lines, different lines. We looked at a lot of different ones. And this is some comparative studies we've done on Aedes aegypti. Here's a map that's now a few years old. I assume that this is much more expanded now, even at this point. There's a little mark on there. I know we have them in Cincinnati. You wouldn't think we had in Cincinnati, but they show up at the end of every summer because we have a tire recycling facility in the city, and they show up and then they're dead by the spring again if we don't see them again. So it's every single year. Give us 10 more years, and I'm willing to bet they may survive there because it's getting a lot warmer. So mainly we were really wanting to compare among different species of this one, of mosquitoes, and there's been a lot of work in Drosophila where they compared sleep levels between different populations, not different species. We wanted to really look at different populations here. Really, in mosquitoes, there have really been only one study. It was Anopheles gambiae, but that was the M and S form, which is now technically two different species. And then there was a little bit on some of the Culex pipiens population, but it wasn't really that great it was a basic comparison. And so we wanted to look at this among different species. And so overall for that, we decided that we looked at the basic rest activity sleep patterns of 17 different populations of Aedes aegypti. And so we got these here, 17 West African populations. We got those from Noah Rose and Lindy McBride. And then we got 12 lab populations in the US. And then we did a whole bunch of modeling, statistical comparison among the different lines. And so here is just the general ones we got from the lines. So this is Noah Rose. He did his PhD down at Princeton, so some people may know who he is, with Lindy McBride. We got all these different populations from him. They have different levels of, they're different from different levels of, they're collected from areas with like different urbanization, different levels of formosus kind of combinations with it. And so you can do some like comparative ones on basically, are they collected from an area with more people? And so it turns out at least for, give you a quick highlight, the diurnal behavior is just consistent among all Aedes aegypti that we looked at. Tell you, we have found one line that's nocturnal now of Aedes aegypti, and we've also found one line of Aedes japonicus that's also nocturnal. So weirdly, we have no idea why that happened. And so we're actually delving into that a little bit more because we think it might be interesting to know. Like that really distinct transition does not happen that often. So one seems to be from the mountains, and another one seems to be like from a suburban area. And we have no idea why for those two. Which one? They're from our field station in Ohio. And so in Harrison, Ohio, we have them. I could go a little bit into this. We think they're feeding on frogs. So we think they made a host transition to feed on frogs. It turns out there's this one site on this one stream. We find them there every year. And that population seems to have made a transition. And we really only see them burgeoning when all these local wood frog and toads are out all reproducing. And we did some PCR, and it looks like they've transitioned to feeding on amphibians in that population. That's a Japanicus, yeah. I know, it caught us, and Clement and Chloe Alejandre at Virginia Tech, we're also working on that with them. They may be seeing a similar transition that's happening there too. So it looks like a potential host shift in them, kind of cool. So again, that's gonna take us a while to finish. We do see a lot of differences among the lines. We do see a general trend, and this is something that probably should be thought of in respect to lab colonies of Aedes aegypti. Lab colonies are almost always more active than what we see in actual nature. And so, and we're not sure why exactly. And then if you look at the general sleep, because of that, inherently they sleep less. We think you're in this nice cushy life. It's changing your behavioral dynamics, and they almost always have sugar and water. They have chronic access to a blood meal. Their lifespan is actually probably generally shorter than some of the potential that you get in the field populations. So we think there's some big differences here. And then the main take home from the paper is we did all these different populations from Noah's group and from Noah's paper, and then we looked at some of the correlations with sleep levels and activity. And really the only thing that popped up is it turns out there's a little bit of the ancestry, which is a Aedes aegypti versus Aedes aegypti formosus, like the comparisons there. There's probably a difference in there for activity. And then it's also a host preference, but those two things are intertwined partially in the statistics. And so there's something going on with ancestry host preference that is making them a little bit more active during the day. And we think it's probably, they're more active during the day because they're probably feeding more towards humans is where the correlation's going. And humans are a little bit more active during that like day period versus maybe like some primates that may be a little bit more active or other species are feeding on. So, but I reached out to Noah a little too late. He didn't have that many lines left. We could have really benefited from like 20 lines here, but by the time we got him, we only had about 7 or 8 lines left. So all right, so this is the summary for this. It was published in Medical and Veterinary Entomology, I think at the end of last year. So yeah, so I still got enough time, right? It goes till, okay. All right, I just looked at the clock and I realized, oh God, maybe I broke. So all right, so the last study I'm gonna talk about is some of our recent work on the dynamics between dehydration and sleep activity profiles. And mainly we did some earlier work on this that showed exposure to dry conditions impacts feeding parameters of mosquitoes, but we were really interested in how this impacts them post blood meal. So, and really some of the general thought a lot of people think about is a mosquito will feed once, and then everybody says, oh, then they won't feed again until after they lay eggs. That's not really true I think, and some lineages will feed more often than others as taking a secondary blood meal. And there were a lot of good studies by Tom Scott and John Edmond where they did some populations in Puerto Rico, and they said a lot of them will take sometimes 1, 2, 3 blood meals per time. And you can see the blood meals in there. I mean, it's pretty easy. If you've ever done that, you can go into your colony and open it up and see how many have taken multiple blood meals. And sometimes it's 1, 2, 3. The most you can usually see is 3. Sometimes you can see like a little remnant of maybe a fourth, but it's usually 3. And so the general thought for a lot of this is, all right, maybe the mosquitoes didn't get enough nutrition as larvae because and so they have to take a couple blood meals, or maybe they had interrupted blood meals on the first one. And so that was the two general thoughts of why this occurs. But we were really interested is, all right, what if mosquitoes, after they blood feed, they like to go blood feed and then go find somewhere that's not really dehydrating to rest and digest their blood meal? What happens if they can't find that, particularly if they get stuck inside the house with you? They get stuck inside your house. Most people's houses are effectively a desert. So, so this is a study and it was mainly by Chris Holmes here, and then a combination of a lot of people from the lab through the years. And so I would say the most important, everybody here is important. I like to emphasize that, but the most important would be Chris, probably Rania and Yanyu, and then Chi helping with some of the circadian aspects with it. And so we designed a lot of experiments where we're going to see is, all right, if we allow them to blood feed, then move them to wet or dry conditions, will they chronically refeed them? And so this we replaces in the paper. It's foot, not hand. So in the review, they said, you technically used a foot, so put a foot rather than a hand. And so I was like, all right, that's fine. We just wanted something as a host. And so we took them, allowed them to blood feed, moved them to wet or dry conditions. And we had different size of cages, ones that were little itty bitty cages all the way up to like the room size 6 foot by 6 foot by 6 foot BioQuip cages that you can't buy anymore. And if you try to buy them, they're like $2,000 now. They used to be like 500. So inflation and that sort of stuff. And so we did different assays here, and mainly we looked at Aedes aegypti and Anopheles stevensii, and it turns out under dry conditions will reduce refeeding. And what on average we see, it turns out you can go through all of this, but I really want to focus on over here is in these bigger cage assays, you'll see them feed about 2 to 2.5 times, rather than under like wet conditions, it'll be like 1.1 times. So they're taking about twice as many blood meals. So when you start thinking about, disease transmission, that's could be a pretty big deal. And we worked on like, what are they using the blood meal for? Like someone said, maybe it's just like egg production, that sort of thing. But really it's not that. We don't see any real difference in the actual egg production. It really just allows them to survive longer in dry periods. It's just an extra water source. You're in a dry environment, you're probably not going to be able to find water so easily. We do see a reduction in the long-term effects, but by just offering them blood meals every couple of days, we can allow them to survive effectively without water for about 20 days. And they will just keep refeeding every couple of days. But eventually they do lay less eggs, but that's because you get weird reabsorption if you can't find a spot to oviposit your eggs. Yeah. If they prefer the water and blood, they will keep refeeding on there, but it's chronic access to blood, to water. So we think like less of them will just blood feed because they're hydrated at that point.
That is, so that if given a choice.
If given a choice, they'll do one or the other usually. We think it's either a preference or it could be the time we're offering them to blood feed. And so we've done some of the preference ones. And if you take them and blood feed them, so we've taken some dehydrated mosquitoes, put them in a wide tube olfactometer, gave them just a humid source or a host source, they still go more to the host source. Like just water with nothing else in there. So just like they're detecting humidity, just water, and then versus like an actual like animal source. socks, old nylon socks from humans. They'll go preferentially to there to, although they've already blood fed before, if they're extremely dehydrated. So rather than the humid source. So I know, and so we weren't sure what's going on with that, and that's from future studies we're working on, yeah. So the last thing is we wanted to see how this impacted the behavior of the mosquitoes. We did some sleep activity assays like before, and the big thing, what it really turns out is about 2 to 3 days after the mosquito, you see an early return to activity. So they start turning back on their activity in a period when they would normally be less active. So it's about 2 to 3 days it starts to occur. They start to be more active, they start to get more agitated, and they start to look a lot more like an unfed mosquito at that point in their activity profiles. So we think it's a return to host seeking. We got a whole bunch of different mutant lines. So like a whole bunch of them, pretty much anybody we could get them from, they just gave them to us or we ordered them. We assumed it was humidity detection. And so we got the mutants for those from Paul Garrity at Brandeis, and it turns out that doesn't impact it at all. So it turns out it looks like it's all the GR mutants, and they're involved in carbon dioxide signaling, and that's why that initial Y2 olfactometer is why we actually did this for this study. And so it turns out we have some follow-up studies with this. It turns out looks like to be an early return to carbon dioxide signaling that seems to occur where they're host seeking again, earlier than you would normally expect. And so that's why they'll take another blood meal. So normally they wouldn't host seek until they laid eggs, but they're host seeking while they're actually developing eggs. I wouldn't call it gene expression. It's probably impacting neuronal circuits underneath. And so that's causing them to become sensitive again towards carbon dioxide. So the gene expression is probably not fast enough to do that. So it's probably more just the neuronal circuits. Yeah. So from this, sorry, I was playing with this on the plane, and I think I got a little bit into the title. We wanted to model how important this was. And this is Yang Yu Zhao. We have a vector biologist modeler in the department. She did this. Don't ask me any questions about this because I kind of know it, but it's not as well as she does. And so really what it happened is if we can look at per day vectorial capacity between the dry and the wet, it becomes much higher due to this. It's plain and simply one of the biggest contributing factors to vectorial capacity, and that's like looking at disease transmission, is just the number of feeding events. You increase feeding events, the entire increase of that is the increase in feeding events. But when you start modeling it, you can look at like the periodic transmission of like Zika on the top right D, and then the survival of Aedes aegypti. And what it turns out is by using this chronic refeeding, it just allows you to bridge dry gaps, like a couple week dry gap, you can keep refeeding. And what it does is it seems to make refuge populations that can more quickly rebound when conditions become more favorable again. It's probably not going to get you through a full dry season. They'll probably use their dehydration resistant eggs to get through this. But like we've had around, I don't know how you guys have been around here. In Ohio, we've had these like weeks of dry periods recently. And the mosquitoes, for a little bit of time, we're seeing them feed more often. Now they're dying because there's no water at all. But we think it can be a mechanism to survive like shorter, like maybe like 2 to 3 week periods of dry conditions. So, okay, and this is the model we have for it. And so it came out in iScience, I think, earlier this year. So it's really, you feed, get in dry conditions, it'll cause chronic refeeding. The reasoning for that seems to be an early return of carbon dioxide sensing and increased activity. And then the multiple blood meals means more disease transmission and particularly higher survival in dry periods. So. All right, so this is pretty much the overall summary on this. I'm going to talk about some of our recent studies. We have some really good evidence overall in 1 and 2 that there's probably some strong sleep-like states in mosquitoes. We didn't even talk about here. We have now RNA-seq on the rebound, and it looks like it's linked to at least 2, if not 3 circadian genes. We have differences among the different lines, which is expected and has been seen in other systems. And there seems to be this interaction between dehydration, sleep, and activity profiles in mosquitoes that seems to alter their post-feeding behavior, and they'll take a secondary blood meal. So from that. And so that would normally be the conclusion, but we just popped out another study on this with Clement from Virginia Tech. We did some work with hemoglobin in the blood meal and wanted to see how this actually impact sleep activity profiles. And so we had regular blood, blood with hemoglobin, blood without hemoglobin, or fake blood without hemoglobin, mosquito snack. Anybody knows what that is? It works as a blood substitute. It's just not great. So we had it with and without, and then just sugar. And overall, what we see is you see issues when you get rid of hemoglobin. They behave weirdly. And in the next one, really the lack of hemoglobin diet causes a reduced level of viable eggs. And so we think without the hemoglobin in the blood, it's really just, they think they're getting a partial blood meal. Yeah, no, it's replaced with the protein. It's the iron from the hemoglobin. So I didn't, in the fake blood, yeah. So it's more of, we swapped it out with the same amino acid level, but so it's really the lack of iron in there. So it's the iron would be from the hemoglobin. So I could have explained it. I just put this in yesterday because we just got it out a little bit ago. So it's the lack of iron. So we actually went in and put other proteins in there to replace the protein lacking from the hemoglobin. So it's the same caloric levels of protein, lipids, that sort of thing. It's just the lack of the iron from the hemoglobin. And the reasoning for that, we can say, is that they've done some radio-labeled studies of mosquito blood, and they'll use about 7 to 8% of the iron from the blood to actually probably generate eggs in other aspects. So we think that's why. And then there's just pretty much almost the egg viability is down to 25%. So we think the difference is due to the lack of iron in there. And then so that's the iron from the hemoglobin. So and then we did aspects post blood feeding with sleep deprivation if it impacts egg production, and it turns out it does nothing. So we deprived them for an entire night, and not significant, not significant, not significant, not significant, not significant. Like the stuff over here, the activity and the sleep levels don't really matter. They're just they're blood fed and then sleep deprived in this case. And so they're blood fed, then sleep deprived. And so we sleep deprive them. It's like the day and a half after the blood meal when they're probably they're still digesting, making a little vitellogenin. We had some thoughts that maybe we could do a longer sleep deprivation, see a phenotype. Problem is if you go beyond this like 1 12-hour window, it starts to get a little, I think, too stressful for the mosquitoes, and then they start to die. But we think it's just too much stress. And so we end up having to do that. So we have this study. It's on bioRxiv now, so if anybody's interested in seeing that. And so in general, so I'm to the end of the talk. And so in general, thank my lab here. It was mainly Clement and then Matthew there with his ice cream there. What you don't see there is his entire top of his ice cream cone just fell off like 2 minutes before that. He was very unhappy. We went to a place in Ohio called Young's Dairy, and we're feeding the goats and that sort of thing. And then some more acknowledgments for some of this work. Sam helped with some of it. Diane and Nicole worked in Clement's lab, and they did help with some of the studies there, particularly the hemoglobin studies, and then some of the studies on comparing the different population. Shichi is now faculty at Georgia. It's either college and state university or state and college university in Georgia, and so he's there. Shovik is now a postdoc at the Tata Institute in India, and so they worked on it as well. And then a whole bunch of the undergrads through the years. And so this is a combination of all the undergrads who worked on the project over the last 5 to 6 years. And so most of them, every one of them now, other than one, is already on at least one. Some of them are on up to 3 research papers they've contributed to. So, and then the last thing, all our funding, NSF, NIH, USDA. The USDA is mainly funding studies on some thrips biology that we've been working on, and then the NSF has funded the Drosophila stuff, and the NIH has funded most of the mosquito work.